Dr. Halder is there. Dr. Purnam Halder is there.
Aditi, I think you can start now.
Yes, it is 6 p.m. sharp, so we are starting.
Oh, yeah.
Okay. Very good evening to you all. Am I audible?
Yes. Yeah.
A very good evening to you all. It is my privilege to extend a warm welcome to all participants registered from different institutions in India and also abroad in today's webinar, Recent Trends in Chemistry in the fields of medicine and industry. Before beginning, I would like to request our Google Meet participants to keep their video and audio muted and refrain from the start presentation mode. And since this is a program entirely based on network, so in case of any network issue, I would also request all our participants to bear with us. If any such issue arises, we promise to resolve soon. Now, this is the second webinar of the Chemistry Department of President's University, and I take this opportunity to thank all the participants for showing that interest and registering at our webinar at these trying times of the COVID-19 pandemic. Today, we are extremely privileged to have our Honorable Vice Chancellor, Ma'am, Professor Anuradha Lohia, for the inaugural session. We are very much honored that she has accepted our invitation to deliver the presidential address in spite of her busy schedule. Thank you very much, ma'am. Ma'am, I would also like to inform you that we have a huge number of participants who have registered from different cities across India and also from different countries around the globe. Today, the 5th of September is celebrated as Teachers' Day in India to commemorate the birth anniversary of our ex-president, Dr. Sarvapalli Radhakrishnan, who was a great scholar and teacher. Upon paying our homage to this great son of India, we will move forward towards our inaugural session. And I would like to request Dr. Anjoy Maji, convener of the webinar, to take over the inaugural process. Over to you, Dr. Maji.
Thank you, Dr. Aditi Bose. Good evening, everyone, and warm welcome to One Day International Webinar conducted by our Department of Chemistry, President's University, Kolkata. Myself, Dr. Anjoy Maji, Assistant Professor of this department. Earlier, our department used to conduct seminar in regular basis, but due to this pandemic situation, very recently we have started conducting virtual seminar or webinar. And recently we were conducted successfully our first webinar, and today is the second. First of all, I am welcoming our Honorable Vice Chancellor Madam, Professor Anuradha Lohia, Professor Gandhi Kumar Kaur, Head Department of Chemistry, Professor Vijayan Das, Department of Chemistry, Dr. Arnav Haldar, Associate Professor, Department of Chemistry, and our distinguished speakers, Professor Dipanjan Pan from University of Maryland, Baltimore, USA, Dr. Vishnu Joshi from Ajanas Inc., USA, and Dr. Pranav Haldar from Novartis, India. Finally, I extend our warm welcome to all participants. I hope this evening will be wonderful and enjoyable with chemistry. Before closing my welcome address, I request all the participants to write the question arises during the presentation only in the chat box. At the end of each presentation, we will bring them up. Also, I would like to request all the participants to keep mic and video mute during presentation. And if any technical issues arises, kindly bear with us. We will try to resume it shortly. Feedback link will be shared at end of the session only. The same will be active for 2 hours from it posted. Thank you and welcome again. Now I would like to request Madam Professor Anuradha Lohia, Honorable Vice Chancellor of President's University, Kolkata, for presidential address. Madam, to you.
First of all, a very, very warm welcome and hearty greetings for Teachers' Day. All of us present here, the faculty and the students, we celebrate this together. And it is my proud privilege to welcome Professor Joshi, Professor Pan, and Professor Haldar to this lovely gathering. So happy Teachers' Day, everybody. And it's a pity we can't celebrate it in person, but this technology is fantastic. We really don't need to be separated from each other. Feels like we are together, and really a lot of work has happened. For example, we couldn't have had our overseas speakers here today easily if they were not traveling. And today we are together, so it's fantastic. I have no complaints about internet and this technology which brings us all together. I do congratulate the Department of Chemistry, Professor Gandhi Kaur, Aditi Bose, Anjay Maji, Arnab Haldar, Vijay Das, and all the other faculty members for putting together this fantastic program. Because I'm sure it must have required a lot of effort, thought, and also coordination to bring us all together. So, well done for this wonderful effort. Before I move on, I'd like you to know, those of you who are not the indigenous members of our department, that this is one of the oldest departments of chemistry probably in the country. And we're very proud of our forefather, Acharya Prafulla Chandra Ray, who was the first chemist in India to think Swadeshi. And he made chemistry, he made it possible at a time when academics and industry were completely separated, divested of each other, to make it possible that chemistry could be commercialized. He was the first person, set up a wonderful company called Bengal Chemical, which continues to serve us to this day. And today, his legacy is borne by each of you. I saw that in the series of the 3 speakers, one of them is involved with 5 or 6 startups in his university. Mr. Haldar is also part of an industry. And so I think the legacy continues worldwide. But we're very proud that we are the sons and daughters of Acharya Prafulla Chandra Ray. And he was honored by the British Royal Society. We have the plaque in our campus. He was one of the first Indians to be honored this way. Coming down from that earlier times, I must tell you that this is a very, very wonderful department of which I personally am very proud. They've gone through very difficult times. And I tell you, I appreciate when the faculty fights with me. That means there is hunger in their belly. There is fire in them and they want more. As a Vice Chancellor, I couldn't have asked for anything better. And I have tried to provide them with the kind of infrastructure they needed. This was a building which was made in the 60s and probably never underwent any renovation or refurbishing till recent times. And it's only in the last 2 years that it required a huge amount of funds, that we could garner the funds and also maybe not satisfy them exactly as they wanted, but at least provide them with a working place where they can think, where they can research, where they can teach. And I think I get many complaints less nowadays, so I think they're a little happy. I'm happy about this. And they've also always been involved in organizing very interesting seminars. So I'm very proud that today on 5th September, they've organized this international seminar and that so many of you have agreed to participate. And for all our 3 distinguished speakers to give up their time, I'm sure it's different time of the day from wherever you are and you've joined us. So thank you, Dr. Joshi, Dr. Pan and Dr. Haldar for this wonderful thing. I also think that our chemistry faculty has been a little secluded from what's happening in the modern world of chemistry. And looking at Dr. Joshi, Dr. Pan's and Dr. Haldar's biodata, I can see that chemistry and biology have almost merged. Both are at the cutting edge of that science which is poised on drug discovery, treatment, delivery, everything that is to do with disease, with pharmaceuticals. And I noticed that one of the speakers is going to talk about pharma as a brilliant career. I also was delighted with the topic on COVID-19 using nanoscience, I think. I forget the third. One was imaging in cancer, I think. So imaging, providing clues, stepping across boundaries and out of your comfort zone, embracing physics and biology and of course mathematics. I really look forward to the day when we won't have separate departments, when we'll be one huge school of natural sciences. because together we serve the cause of humanity. And our chemistry professors, faculty are very ably suited to. They've also worked under very trying circumstances in different parts of Bengal and the country where virtually nothing was possible. Our head of the department, Professor Gandhi Kaur, is one such shining example who's published brilliantly from a rural college. The treasure he has garnered in his lab of synthetic compounds which he's made, I wish I were 20 years younger and I could work with him to do what he has discovered. So with these few words, and I also look forward to a very, very bright future for our faculty like Aditi, Anjoy, Arnab, Sumon, and others. I'm sorry, I can't recall everybody's name right now. Now you know I'm aging. So if any of you discover an anti-aging memory formula, please, I'll be your first guinea pig. Happy to take it. I hope you enjoy everybody's talks today and inspire each of us to go forward and embrace this kind of fusion and bring out new solutions to pressing problems in the world today. Thank you for inviting me to inaugurate your seminar, and I wish you all the speakers and the listeners and the discussants a great amount of fun in the coming few hours. Thank you very much.
Thank you very much, madam, for your enthusiastic speech. So thank you again. Now I would like to request Professor Gandhi Kumar Kaur, head of the department, for inaugural address, and also I request Gandhi Kumar Kaur for chairing the session for the first lecture. Gandhi Kumar Kaur, over to you.
Am I audible?
Yes.
Okay. Good evening, Honorable Vice Chancellor Madam, Professor Anurag here, distinguished speakers, Professor Dipanjan Pan, University of Maryland, Baltimore, Dr. Vishnu Joshi, Associate Director of Research, Agenus Incorporation, Dr. Pranav Haldar, Head, Strategic Resourcing, Novartis Sandoz, participant faculty members, research scholars, students. On behalf of organizing committee, I heartily welcome you all in this webinar. This is our second webinar in last 2 months. Truly speaking, Department of Chemistry, Presidency University, though looks young of age of about 10 years, but it bears a heritage of about 146 years from Presidency College. Thus it is blooming from a new bud of an old tree. We strongly believe that it will spread beautiful fragrance all over the world in near future through our products, that is the students, our research activity, our teaching activity. There is a such change in national educational policy. Nowadays, whatever we are doing through our research or teaching must have an impact in development of our industry, development of our society, development of our economy, development of humanity. Thus, industry-academia interaction is getting more and more interaction day by day, and it is very important. Current academicians and students must keep it in mind. Simple theoretical knowledge would give you no credit today. Your teaching, research must contribute in development of country, in development of self-dependence, in fulfillment of the need of the society for food, clothes, medicines, etc. During our research life, a large synthesis of a complex molecule using sophisticated reagents, sophisticated procedures was a very good research work. Now the scenario has changed. Role of material science, role of polymer science, life science is getting importance in chemical research work. It is hybridized now. A research output must have an application in industry, must develop economy of the country. That's why academia industry interaction is necessary. In this webinar, you will enjoy the flavor of the lectures from academic world as well as industry. you will know how the changes are coming out in industrial research and what are the necessary criteria to get a job in industry. And that's why we have arranged this webinar, Recent Trends in Chemistry in Fields of Medicine and Industry. Hope you will enjoy the program very much. Thank you all. Can we go for the next session? Can we go for the
Yes,
next
sir.
session?
Yes, sir. Yes, sir. You move forward and you please share this session.
Okay, okay. Thank you, Dr. Chatterjee.
Shashi.
Okay, our next session, that will be the first lecture by Professor Dipanjan. He will talk on multidisciplinary biosensing approaches to COVID-19 from molecular targeting to digital detection. Professor Dipanjan is personally known to me for more than 25 years. Actually, he did the PhD degree from IIT Kharagpur under the supervisor Professor Jayanto Kumar Roy, who is also my supervisor. And also, I would also like to mention Dr. Pranav Haldar is also did the PhD degree from the same laboratory. So we all are known to each other from long time back. Professor Dipanjan did his MSc and BSc degree from Vidyasagar University and then completed his PhD degree from IIT Kharagpur, and then he moved to State University of New York for his postdoctoral work. And later on, he joined first the University of Illinois Urbana-Champaign as associate professor and associate head of the department in bioengineering and material science and engineering, and later on he moved to University of Maryland, Baltimore, where he is currently working as a professor in diagnostic radiology and nuclear medicine, pediatrics, and chemical, biochemical, and environmental engineering in the Department of Maryland, Baltimore School of Medicine and University of Maryland, Baltimore County. His expertise in nanomedicine, molecular imaging, drug delivery, biosensing. His research uniquely merges the fundamental chemistry, biology, and engineering to bring solution to today's healthcare problem. Dr. Pan has mentored more than 15 postdoc scientists, 14 PhD and MS students, and more than 30 undergraduate students. Dr. Pan has published more than 200 papers in high-impact peer-reviewed journals and conference proceedings. He received funding of multi-million-dollar from NIH, NSF, DoD, American Heart Association, and other private sources. He received lot of awards. Few notables are Nanomaterial Research Award from NML 2016, Young Innovator Award, Biomedical Engineering Society 2017, Dean Award for Research Excellence 2018. He is an elected fellow of Royal Society of Chemistry, American Heart Association, elected fellow of American College of Cardiology. He is associate editor of Wired's Nanomedicine and Nanobiotechnology, editorial board member of Scientific Reports, Nature Publishing, editorial board member of Molecular Pharmaceutics, American Chemical Society. With these few words, I would like now to invite Dr. Pan to deliver his lecture. Dr. Pan, please. Dr. Dipanjan Pan?
Yes, I'm talking. Sorry. Thank you. I was trying to navigate my screen. So
Okay,
thank you for.
Time. I think we have.
Yeah. Thank you, sir, for this kind introduction. And again, I'm trying to navigate to my PowerPoint. I think it is there. All right, so good afternoon, everyone. Thank you so much for inviting me. This is a real pleasure. And today is Teachers' Day, and I could see that at least two of my teachers are in the audience today. One is my PhD advisor, Professor Ray, and also Professor Kaur, who, as he mentioned, that we have been associated for more than 25 years, and I know him from my undergraduate studies. So what I am today is, I think a lot of credit goes to these individuals. So thank you so much for being a great teacher and mentor to me for over the years. And again, thank you for inviting me to give a talk. I was traveling. I couldn't. This is again a great pleasure for me to be here, and this is kind of becoming a norm to give a webinar. This is probably my 10th webinar to present over this COVID-19 situation. So. With that brief introduction, I just want to say that I recently moved from University of Illinois, Urbana-Champaign, where I was part of bioengineering, material science, Beckman, and their College of Medicine. And right now I have a dual appointment between the engineering and medicine at University of Maryland, and I'm a full professor in both medicine and engineering. So today we're going to talk about, when I received this invitation and looking at the topic of the webinar or conference, you can say. I was thinking about what to talk, and because we do a lot of different kinds of stuff. But then I thought about talking about our recent work on COVID-19. So we're going to talk about a few biosensing approaches for COVID-19 and how we're really doing from molecular targeting to digital detection. My disclosure of NIH grants, and I'm founder of 3 startups, and I also have a consulting agreement with a few industrial companies. So this is my group. We're located in 2 locations. One is in the Baltimore County campus, in the Interdisciplinary Life Sciences building, and on the 3rd floor where we have kind of a wet lab, in vitro studies and ex vivo studies. Also, we have small animal studies facilities in the basement. And then we also have another location in the downtown Baltimore, in the Health Sciences Facilities 3, over the 8th floor, where we have a wet lab, in vitro studies happens there, and also some large animal studies, for example, rabbits and pigs. And I also direct a nanoparticle fabrication and characterization core, and also lead Department of Radiology's imaging probe development and characterization facilities. There's seamless interaction between these 2 different locations, different institutions, students and postdocs that come and interact with each other between these 2 locations, and we have absolutely. I'm very proud that we have this collaborative relationship between these 2 complete separate entities. One of the startups inside is located at BW Tech at UMBC that develops sensors for detecting eye injuries, and the other startup, Kilocyte, that develops artificial blood that is located again in the in the University of Maryland Biopark and the downtown location. So kind of two startups. They're based in two different locations, and the third startup, Vitruvian, that is part of the University of Maryland Biopark at the downtown location. So the very multidisciplinary group of researchers. What we do, we kind of merge chemistry, biology, and engineering all together to bring solutions to today's biomedical problem, and these are all very highly translational. So we're heavily interested in basic science, but at the same time keeping in mind that at the end of the day, if our discoveries do not reach human, then there is no point of doing research. So I call it molecule making and device making. I've been trained as a synthetic organic chemist, and then the research evolved with from my postdoc and other experiences. And so right now I call myself a contaminated chemist. Here's one example that you're looking at, molecular imaging of nascent biological receptors. So what you're looking at is a fundamental chemistry, but at the same time, cutting edge imaging. So, for example, what you're looking at here is the internal structure of a rat, and that has been delineated by different kinds of contrast agents. And we're looking at it by X-ray imaging, but you probably have seen X-ray imaging in black and white, but now we're capable of looking at it by in color. So different colors, and that kind of gives a lot of different kinds of information. So we're extremely, we're at the very forefront of this molecular imaging research, and we're combining something we call a photon counting computer tomographic imaging. So on the left side, you're looking at a rat image of the internal structure. On the right side, you're looking at again a rat structure, but then the circle, that the white circle that you're looking at is basically a bone crack that has been detected by some nanoparticle that are binding or homing to those tiny bone cracks that is otherwise not detectable by any of the imaging techniques or by the doctors. The same thing here, you're looking at the bone structures. Again, we also work on drug discovery, ligand screening, and targeted delivery. We are, at the end of the day, our core expertise is chemistry. So what we do is, taking the cues from fundamental chemists, we try to look at the protein structure. We try to identify different kinds of new molecules, or even repurpose some of the existing drugs, and putting it as a nano formulation and try to deliver it to the disease site by a safer means. So here on the left side, you're looking at a cartoon of a nanoparticle that is seeking out cancer stem cells and delivering drug to the cancer stem cells. On the right side, you're looking at a molecule, a heterocyclic molecule that is binding to the inner groove of something which is known as a retinoid X receptor or a nuclear receptor, RXR. And then third front of our research is biosensing. So biosensing is something, it is kind of a relatively newer area of our research, and we have been doing it for the last 6-7 years, but we have been extremely productive in that area, and that's one of the reasons why I call it molecule making and device making, because this is exactly where our engineering fundamentals comes in, and a lot of electrical engineering and electric communication type things that we bring in. So here you're looking at a handheld device, and the design is based on hummingbird, what we're doing is trying to this device is being developed by one of my startup companies, Insight, that shows that there's a tiny bit of a sensing platform, that red circle that you're looking at, that is capable of detecting tear film. That means the tears that are present in our eyes. And then by looking at some markers that is present in the tear film, we can tell if the person is going through some kind of a trauma, or even whether the person is recovering from, say, cataract surgery or other kind of eye surgeries. So there's some very in very short, these are some of the biosensing, molecular imaging, and drug delivery things that we typically do. So we call it translational nanomedicine because nanoparticle is our bread and butter. Basically, we develop every day. Many nanoparticles has been developed in my laboratories, and we have been publishing it, all these things in high impact journal, PNAS, Anglichem, JACS, SS Nano over last 10-15 years. And again, we talked about this multi-scale molecular imaging, different kinds of imaging techniques we use, MRI, computed tomographic imaging, FTIR imaging, Raman imaging, photoacoustic, and fluorescence. New and repurposed and translatable nanotherapeutics. We target transcription factors, oncogenes, signaling pathways that looks at inhibition of stem cell stem cells in cancer cells, and other exploratory therapies, for example, metabolic therapy, pH therapy, nuclear receptors, and all kinds. One particular example that I just wanted to, I'm very proud of this work, is that we're trying to generate nanoparticles inside cells. So one of the major drawbacks for nanoparticle is the toxicity, and that we all know that. And so in this particular study, what we're trying to do is we're trying to generate nanoparticles inside the cell by utilizing the cellular machinery. That means if there are some proteins inside the cell, then those proteins will be capable of reducing some molecules. For example, if you're using gold chloride, then gold chloride will be reduced by the intrinsic or endogenous proteins that are present in the cells, and they'll be generated inside the cell. So the nanoparticles, really, they're not really they're not really systemically administered. They're not really traveling through the blood vessel. So the chance of having toxic effect is really, really less. So what we demonstrated here by in vitro, A, B, C, D, all you're seeing the in vitro results, that intracellular in vitro reduction that is occurring en route, and the gold nanoparticles are forming inside the cell. So that means if you're targeting a particular cell, say for example a cancer cell, then the particles are being formed inside the cell. So the chance of having any kind of a detrimental effect to the surrounding cell would be really, really minimal. And we also proved that, and there is a typo that's it should be in vivo reduction on the left side, on the right side. What we're seeing here, what we're looking at here is that in a mouse model, we're showing that the particles are being generated, and we can apply laser and treat those the tumors. Treat those tumors by applying the thermal therapy. And this paper is actually, getting published in Nature Communications, and this is going to be out in a few weeks from now. So with that, I think this is pretty much kind of what we do. We do molecular imaging, therapy, and biosensing. Now, with that, kind of brief introduction of what we really do, this is something new, COVID-19, and as we know that COVID-19 kind of changed the way we look at our life, the way we lead our life. I mean, I wouldn't think about giving a webinar talk if this was not because of COVID-19, right? So in a sense, COVID-19 changed the way we live our life, and this is now the new normal. So this is truly a global pandemic. The first case reported in China back in 2019 December. Now we're slowly learning that there's probably at least 4 or 5 months before that the actual pandemic started, but we've probably not been informed at that time. So 11th March 2020, WHO, they recognized the outbreak as a pandemic, and at that point, the whole, all the countries, over the world, they're all we're shut down, and we're under the lockdown period, and the labs are closed, schools are closed, offices closed, everything is life kind of stopped. So far, I think this is the number from today, 26.6 million worldwide confirmed cases due to coronavirus. And in the US, I think this is a little bit of old data, June 2020, 2 million cases. And 875,000 deaths worldwide. This is today's number, with 188,000 deaths in the US. And WHO has appealed for global mass testing, with a demand that is estimated to be more than 600 million tests, and with a combination of genetic and 500 million rapid tests. So this is truly a global pandemic, and at that point, my lab was closed for a month or so, and then we realized that we really need to respond to this global pandemic because this is our responsibility. So before we go and talk about what we have done, let's talk about a little bit about the genetic makeup of SARS-CoV-2. So if you look at the genetic makeup, then you will see that there is a 96% of the nucleotide matching with bat coronavirus, 91% match with pangolin coronavirus, and the SARS virus that happened a few years back, there's an 80% of genetic makeup match. MERS virus that has 55% match, and there's only 50% match with a common cold virus, so like a flu virus. The virus by itself is really tiny, SARS-CoV-2, only 30,000 base pairs that makes up this virus genome, and there is a spike protein on the surface that binds to ACE2 inhibitor, the proteins on the human, and that is a result of a natural selection. The virus, and this is probably for good, it undergoes some mutation, but not really that much. And so occasional mutation happens, and the major culprit is the spike protein that undergoes the mutation, and then that will allow the virus to kind of partially escape the vaccines or any kind of hard immunity that are being formed in humans. So $9 billion, and I think this is even more. The market opportunity is really, the diagnostic market opportunity is $9 billion, and that is by 2025. And why is that? And this is not really $9 billion, but this is going to be even more, because we know that this is COVID-19, but there is a high probability that there will be a COVID-20 or COVID-21, because these viruses are changing their mutation, and this is coming from, most of this, the origin are really from wildlife, and the way we're not being careful how these wild animals are being treated, and we're taken to the meat markets, and in how we're handling those things, I think there will be another pandemic coming. So we really need to be prepared for before the next thing happens. If you look at some of the analytical approaches that are being used today, then you will find that the first column, that looking at QRT-PCR, which is really the gold standard, every big hospital, large hospital, every diagnostic center, large diagnostic center, they should have a thermocycler or a QRT-PCR device to detect these viruses. So there is no, hands down, there is no parallel in between these with the RT-PCR and any other tests. The sensitivity is more than 97%. Specificity is almost 100%. Limit of detection is really copies per mil 100 to 1,000. At test time, that is where we are really struggling, because a sample to answer time from the time when a person submits the sample, either saliva or nasal swab, and to get the result back, it takes about 4 to 6 days. You can cut it down for some faster labs. They can cut it down to 1 day, but still 1 day is really high, because by that time, if the person is not really quarantining himself, then the person is really infecting all the other people that are coming in contact with those. So that's a real trouble. And also the price point is really high, because you really need to have a big RT-PCR machine in your facility. You need to have a skilled person who knows how to run this system, and so that all that adds up, and that brings us to the point that we really need something which can be deployed, which can be utilized at a point of care setting, where, say, for example, like we need to send our kids back to the school. They cannot really do their classes from sitting at home. We need to send all the kids back to the school. We need to send all the students back to the college, and everyone needs to be kind of leading their life. So how can we really do that? And for that, we need something that can be done get the result very quickly without requiring expensive equipment, but keeping it the sensitivity and specificity that is required, and the virus that is causing this pandemic. Some of the competitors, so most of the FDA approved, or and you must be reading all the newspapers. There are multiple tests coming out, and most of them are serological. That means you plasma, and you try to determine the antibody that is present in the after an antibody like IgG or IgM. The problem with that approach is that this is the effect of the virus on our body. If there is a foreign entity fight against that virus, and that will be generated, and that will be developed inside our body. So we are trying to determine if the body is, and I'll tell you in a minute why that approach is not really come up with some point of care test or rapid test, as we can say, and few academic group, of course, my group. So let's see where we are. So now, what is the characteristic infection progression in a single patient? So if you look at it, then you will see that there is almost 5 days, or 3 to 5 days of latent period. That means the 5 days of this incubation and latent period where the person has been infected, but there is no symptoms, and virus is not really doing anything. It is really sitting there and not really doing anything. After that initial five days, the patient becomes symptomatic, and some of the patients even remain asymptomatic, and there are different reasons why that happens. So literally for 0 to 7 days before actual manifestation of the symptoms, there is nothing happening. And that is the most important time, because if we really need to identify if that person has been infected by the virus or not. So if you look at this is kind of the incubation and kind of the roadmap of the virus inside our body. then you will see that until week 2, there is no antibody generation inside our body. So almost 7 to 14 days it takes for our body to respond to this virus. So if we are really detecting at that point, we really don't need that, because by that time the person is already has been infected by the virus. They're generating the antibodies, and there is the person is already has been infected with some symptoms coming out, and there is no real reason for detecting whether the virus has been there in his body for last 14 days or not. So we really need to identify this virus at a very early stage. So early detection is the key, and you can see this plot that at the presence of SARS-CoV-2 viral RNA in swab or saliva samples, if we can do that at 0 day, that means that as early as the first day of the infection, that's where the importance comes. So how can we really do that? We can only do that if we can identify the RNA, if we can come up with an RNA-based detection technique, because we really cannot do that by antibody, because antibody is not going to be generated until 2 weeks. So early detection is the key, but at the same time, we need to be doing that by limited resource use, easy to operate something, highly sensitive. We need to get to the lowest detection range possible to match with RT-PCR. Should have high specificity, and it should be able to differentiate between other coronaviruses or even flu viruses. So how can we really do that? So before we do that, we need to know the enemy, right? So that's SARS-CoV-2. You're looking at a cross-section of the SARS-CoV-2 where you're, this is the spike trimer, that the S protein that is on the surface, that actually binds to the SEE2 proteins on the host cell, and that is primed by another protein known as TMPRSS2. And then there are other proteins that are important. So for example, membrane protein, that is M protein, nuclear protein, that is N, and our interest is in the nuclear protein, or NG, because that particular protein does not really undergo mutation there. So that is kind of mutationally stable. So we really wanted to go after the proteins of the gene that is nuclear protein, because that does not really undergo mutation. If we go after spike trimer that undergoes mutation, that means if we're developing a test for US that may or may not be applicable or equally effective in India or in the other parts of the world. Because when we started working on this thing, the first sample that we received was from Nigeria. Then we got the sample from Wuhan, China, and then from US. And what we're looking at is that for the spike trimer, really it undergoes mutation, albeit slowly, but it undergoes mutation. Going after engine is the right approach because it really does not undergo that much of mutation. So it is mutation stable. We're going after this nuclear target, and if we can find a particular sequence, then we really can develop a sensitive test. So this is a design principle. What we're doing here is we're coming up with oligos, and oligos that target, or antisense oligos that targets engine. And our approach utilizes a very unique front location binding and end location binding oligos. So we have two ASOs that are binding the front location of the end protein, and the two ASOs that are binding to the end location of the end protein. Why? Because we want to form a claw-like binding so that it has a very classical molecular recognition, right? So a claw-like binding so that it has a much more tighter binding. It doesn't let it go, and when it binds so strongly that the response will be really, really strong. So the ASOs were developed in my lab by Dr. Moitra, who is a research scientist, and he was in Dr. Shantanu Bhattacharya's lab in cultivation science, and then before that he did his PhD from IISc Bangalore. We recently published this work in SS Nano. So this is the backbone structure, 3D structure of the antisense single-stranded DNA. And so what we're doing is we are relying on the fact that, and we wanted to go with something simpler because we wanted, again, this is all for translation that quickly can be translated to human. So what we did, we conjugated these single-stranded DNAs, or you can call it antisense oligos, to the gold nanoparticles by a chemistry. Now, you can see that this is the first TEM image, the transmission electron microscope image, that see that all these particles are beautifully dispersed. There is no aggregation, nothing. Now, the moment the SARS-CoV-2 engine comes, there is a red shift in absorbance, and there's a 40 nanometer red shift. What happens is that there is a rush of these particles coming together, binding to the engine, both in the front location and the end location. And as a result of that, all the gold particles are now aggregated. Now, the gold particles, we know that those are plasmonic particles. So based on their size and shape, they will change how they absorb the light. So based on that, they will change their color. So you can see these clustered gold nanoparticles, after binding to the engine, they're forming a cluster. And that's the basis, that's the hypothesis, that the moment this gold nanoparticle will be clustered, they will form, because of the absorbance, and as a result of that, there will be a change in the color, and that can be appreciated by naked eye without even requiring a spectrophotometer. And that's what's happening. You can see that this is approach 1, plasmonic detection, where you're seeing that the SARS-CoV-2 viral RNA is being incubated with the gold particle, looks blue, purple, and then the moment you are really incubating it with the viral RNA, 5 minutes incubation, the color changes to blue, and then you're using RNase H at 65 degrees centigrade. That's an optional step. The incubation is 5 minutes. It precipitates out as a blue solid. So very simplistic, inexpensive, only 10 minutes after RNA isolation. The detection can be made quantitative if you connect it to a spectrophotometer. The sample volume that is required is only 3 to 5 microliter of RNA, so very, very small. And it can be integrated with lateral flow assay and potentially be integrated with something we call it a loop amplification method. And also potentially be tagged with a color changing mobile app. We published it in ACS Nano 2020. That was the first report of viral, SARS-CoV-2 detection by plasmonic and by any means, actually. And we received NIH funding, 2 NIH funding. That was pretty neat. And then immediately we moved on to something which is known as an electrical chemical detection. That means we were thinking that this probably cannot be done by, think about this way, that this technology is being given to our parents or our grandparents. They really cannot do it. So how can we give something to them that they can use? So then with that kind of concept, we started developing something kind of like a glucometer, like at home glucose testing method. So here for this, the concept still remained the same, the gold ASOs, gold particle, but instead of having a plasmonic detection, now what we have an electrochemical detection. So we have graphene surface. Then on top of that, we have this contour-based microelectrode. And on top of that microelectrode, we're immobilizing these gold particles with ASOs. The idea would be that the moment we're incubating the viral RNA, there will be a strong binding and a disruption of the electrical charge between the graphene and the gold nanoparticles. As a result of that, there will be a change in the resistance or the impedance profile of this the graphene conductive layer, which can be expressed as a voltage against the time, and then we will get a response back, and that can be processed by a software. So this is like a true real-time detection, less than 5 minutes for a stable response, highly quantitative, and the detection sensitivity reaches about femtomolar, and sample volume is only 2 microliter. The sensor will be disposed, so we have a razor blade model, and potentially can be integrated with lamp and other things, and works like a glucose meter. So here you're looking at an ACM image of the scanning electron microscope image of the sensor by itself. This is actually under review with a major journal, and should be, well, we received the comments back, we're responding back. The work was done, with Dr. Moitra, by one of my grad students, Maha Al-Afif, and the research specialist in the lab, Ketan Bige, who has been with me for the last 5 years. So here is the response that you're looking at. That, and I just want to kind of point to, I think I'm, how can I hide this thing? Okay, so you're looking at on the D, the plot D. If you look at the plot D, then you will see that the response of the voltage with SARS-CoV-2 is really dramatic and statistically significant. It's 2-star in comparison to what you're looking at here for MERS-CoV, which is a MERS virus, that is almost nothing. So highly sensitive detection from viral RNA. Now, we took some patient samples, and now we tried to test whether this is working or not. So nasal swab was collected from human patients, and or even saliva. And as you can see here, that the plot A, the red plot is really for the normal sample, and the blue is really the positive COVID-19 sample. So with COVID-19, immediately the sensor sees the COVID-19 RNA. There is a sharp rise in the change in the voltage, and that is taken out by the software, and that has been plotted, and we can get a quantitative response back from it. And in fact, we started in many patient samples, and the result has been absolutely fantastic, with a detection limit of 6 copies per microliter, which is at par or even lower than some of the RT-PCR values. So we're extremely happy with the finding. Approach 3, hyperspectral imaging. So what is hyperspectral imaging? A hyperspectral imaging is, hyperspectral is nothing but it's like a human eye. Like when human eye is I'm looking at outside the window, and I'm looking everything as in different color. The trees are, the leaves are green. Sky is blue, and all those kind of things. So hyperspectral is really an imager that acts like hundreds of spectrophotometer in parallel, and that is capable of generating a spectral curve for each individual pixel that is present in a particular scene. It could be a tissue, it could be a cell, it could be anything else. So we wanted to use this unique technique for detecting SARS-CoV-2. Long story short, I'm not really going into details, but so far, this is by far, and this is so far unpublished, so a little bit confidential, although we have patent protection. What we're seeing that this is by far the most sensitive detection technique for SARS-CoV-2 virus. So same concept, the moment these particles are really coming in contact with the viral RNA, they're aggregating, and as a result of that, there is a sharp change in the hyperspectral for these cluster of nanoparticles. And as a result of that, this is a microscopic-based technique. So the result, the detection sensitivity can be reached up to 1 zeptogram or zeptomolar. So really sensitive, and the results can be obtained instantaneously after the RNA isolation. So it's not even minutes, you can get it in 2 to 3 seconds, the result. The only caveat is that you need to have the microscope. So this is not really for at home, but this is for a hospital-based, platform. So some of the COVID-19 positive and negative samples, you can see that the negative samples are red. That is under that blue dotted line. That's the threshold. And then threshold area under the curve. And then green dots are all the COVID-19 patient samples. So here you're looking at the 59 COVID-19 patients. The positive, we really, well, I thought I changed it. Okay, maybe the accuracy is 100%. The sensitivity is really 98.9%. This is a typo. And the specificity is 100%. There is no cross-reactivity with flu virus or MERS-CoV virus. So we were extremely happy with our outcome. With the negative COVID-19 patients or healthy subjects, we analyzed, this is old data, but at that time we analyzed 30, and 30 were identified as negative. There was no cross-identification, false identification. And then again, we started utilizing a lot of analyzing a lot of samples, and this is my COVID-19 team, and Dr. Moitra and Dr. Ray, who is working as a postdoctoral associate in my lab, and Ketan Dike and Maha Lafif. They're all working very extremely hard, and as you can see here, there's some kind of representative examples of sample RNA from COVID-19 positive samples, and the negative samples, they remained only red, or purple, you could say. But the COVID-19 positive sample, they turned blue immediately after the addition of our reagents. So in brief, this is a Pond Lab approaches for doing the COVID-19 detection. There are three approaches that you're looking at. One is the visual, that is plasmonic based. The second is the electrochemical, and third is the hyperspectral. All of them are based on fundamental chemistry. There's highly specific antisense oligos for engine and plus the gold nanoparticle platform. Now, on the y-axis, you're looking at the limit of detection. Of course, the limit of detection for, as we talked about, for the hyperspectral, it's the maximum. It's or the lowest, I would say. So the zeptomolar. But electrochemical is a femtomolar, and it goes up to like 6 copies per microliter, which is pretty close to, or even lower than for some RT-PCR technique. The visual plasmonic test is not really that sensitive, but at the same time, this is really meant for at-home use. So if you're looking at a true point of care at-home use, you really cannot expect a RT-PCR level sensitive detection. With that, I'm just going to end here saying that my lab, we're interested in doing science that brings us from symptomatic medicine to preemptive medicine. We really don't want to wait for the symptoms to start. We want to see whether the disease can be stopped before even it happens. So we uniquely combine molecule making and device making. So in this case, we are making the antisense oligos, and we are making the gold particles, and then we are combining it with an electrochemical platform, which is making a device platform. Early detection at a molecular level, we're really targeting engine. That's a molecular target. Directed safer therapy, we really didn't talk about that in today's talk. Safer affordable therapy, we didn't really talk about. Our translatable solution, we are seeking FDA approval. At this point, there are at least 10 companies that are trying to license the technology. Some within the U.S., some from outside. We are, University Tech Office. We're not really, they're taking the lead on that. I also started the spin-off Vitruvian Bio for developing the plasmonic test. And but none of these things possible without having an absolutely dedicated team of researchers. This is kind of an old lab picture from Illinois. Extremely hardworking individuals that I'm so proud of, all of them. funding from NIH. We received 2 funding for COVID-19 and just for COVID-19 in the last month or so. And NSF, DoD, Children's Discovery Institute, American Heart. Without the funding, it's not possible. Collaborators all around the U.S. and outside. Mars Bioimaging in New Zealand, we collaborate on the Fulton Counting CT, and Philips Germany. My startups, their CEOs and everyone in my startups, the employees. And finally, thank you so much again for this is. It's been my pleasure to present to you, and I'll be happy to answer any question. Unfortunately, I have another commitment right after this, so I really need to leave, and I already told it to Aditi. So if you can ask me any questions right after this, I'll be very happy to answer your questions. And thank you so much again.
Thank you, Dr. Fan, for the completed lecture. The matter is now open for discussion, and I request Professor Dr. P. Sasikumar and Dr. Anjay Maji to conduct this discussion session.
Good evening to all, and it is nicely. There are a couple of questions I am taking from here in the Google Meet. So, which detection approach can be used as cost-effective commercial technique?
Sure, yeah, that's a great question. So all of them actually, in fact, the plasma test we're right now at the laboratory scale, where we are really developing sensors, say 20 to 50 sensors at a time. Our cost is around $9, and that can be, when we start mass producing it, can be brought down to $1 or $2. So really, I mean, the cost of the whole thing is really not that much. This is we are keeping in mind that this cannot be a heavily costly procedure. For the hyperspectral, I think that's going to be the most costly technique, because someone needs to have a microscope in their facility. So it is like as good as having an RT-PCR. So that is a little bit more costly, but other than that, the plasmonic or the chemical, they're very inexpensive techniques.
The next question, sir. How much time does this virus takes for genetically change?
That's a very good question. So it takes about 2 weeks for the virus to undergo a mild mutation. It's slower than any other viruses known. So that's a good thing, but wait, it really does change. So over 2 weeks period of time, it's changing a little bit. So with that, I think if we're really going after an approach with S protein or antigen-based test, you're really not going to have that specificity for these. So you're going to get some false positive or false negative results.
Now I request Anjay Maji to ask few questions.
Okay. Thank you, Professor Pan. So there is a couple of questions from YouTube chat box. So
sure.
Sudip Ghorai is from Bhava Atomic Research Center. He's asking, can you throw some light on how the agglomeration occurs when COV2N gene comes in vicinity of AU nanoparticle, gold nanoparticle?
I can definitely throw light, because this is no pun intended, because this is a plasmonic shift, right? So absorbing light is is everything. So yeah. So again, I think like the way I was trying to explain that gold is it's a plasmonic material. So what happens is that, and long story short, in a very simple words, the gold changes the way they absorb their light based on their size and shape. So when you have a spherical gold nanoparticle, so for example, the gold nanoparticles that we're really using here, it's 20 nanometer in size. So if you have a 20 nanometer size and if you have a 100 nanometer size, they absorb light differently. So, and that was the hypothesis that we had, that the gold particle would be, and this is like a classical guest chemistry, right? The gold particle will be carrying some guest molecule, and then we're relying on the host, that means the engine that is really waiting for to capture the guest. So the gold particle with the oligos, now they're going, and then the moment they find the perfect the host, they will just bind to it. When that happens, it's not one gold particle. There will be like absolutely thousands of gold particles. They will be coming and they'll be binding to the engine. As a result of that, the obvious, that's, this is the moment the clustering happens, there will be precipitation or agglomeration. As a result of that, that agglomerated particle would now act as a single entity. So they will absorb light all as a single entity. So there will be a shift, and as you can see that we already talked about it, there is an absorbance shift of about 40 nanometer, and that's good enough for us to appreciate the change in color from purple to blue.
Okay, so one more question that is asking by Doipan Chaudhary. The event of mutation is considered to be random. So what makes the nucleoprotein resistant or mutation?
Correct. So the nucleoprotein is something that is a nuclear target, right? So it is inside the virus. So it is really not prone towards not prone to mutation. It is not outside. It is not really on the surface of the virus. It is protected inside the virus. So the virus capsid and the bilayer and everything is really protecting that engine from being, to undergo any kind of mutation process. So that is really not mutationally prone. So it is not really going through any mutation process.
All right. Thank you so much. And I'll hand over it to Dr. Aditi Bose. Over to you, Aditi.
Now we are at the end of the first technical session, and I would like to thank Professor Pan for your exceptional lecture. We are truly enlightened by your lecture, and thank you very much, sir, for being with us. And I would also like to thank Professor Kaur for chairing the session. And now we will move to the second technical session. And at this outset, I would like to invite Professor Bijon Dash from President's University to chair the session and introduce the audience to the speaker.
Thank you so much. Thank you so much. Thank you, everyone. Bye-bye.
Yeah. Thank you, Dr.
Bye, sir.
Thank you, Dr. Thank you, Dr. Bush. Am I audible?
Yes,
Can you
sir.
hear me?
Yes.
Okay. Good evening. First of all, on the occasion of the Teachers' Day, I would like to express my gratitude and honor to the teaching community for their indispensable services to the mankind. The next lecture will be delivered by Dr. Vishnu P. Joshi. He received his BS in Biology and Chemistry in 1999 from Humayun University, Nainital, and his MS from Tribhuvan University, Kathmandu, Nepal in the year 2002. He obtained his PhD degree in Bioorganic Chemistry from Inha University in South Korea in the year 2009. During his doctoral research, he developed various types of peptides for monitoring vital cellular processes. Dr. Joshi pursued his postdoctoral training during 2009 and 2012 at the University of Michigan, Ann Arbor, in the United States, in the field of gastrointestinal cancer research. He then served as a faculty member at the University of Michigan Health System from 2012 to 2015. Dr. Joshi co-authored in over 50 papers in several international journals. He has 8 patents, including US and international, to his credit. He has at present been supervising more than 10 funded research projects. He is currently working as an Associate Director of Research at Agenas Incorporated USA, a clinical stage immuno-oncology company. The title of his today's talk is Molecular Imaging Strategies for the Early Detection of Gastrointestinal Cancers. So may I now request Dr. Joshi for his deliberation.
Thank you. Good evening, everyone. Thank you for inviting me to share my research. And today is kind of like very special day and happy Teachers Day to all of you. So now I'm going to share my slides. Can everyone see my slides? Can everyone see my slides?
Yes, Dr. Joshi, it is good.
Okay, thank you. So, first of all, thank you again, especially like thank you to Anjoy. We were kind of colleague like long time back when I was doing my PhD, and he introduced me to your department, and is like very pleased to present this work in front of all of you. So today I'm going to present my work from my previous lab, where I was working in the molecular imaging, especially for the management of the GI-related cancers at University of Michigan. And I have like included disclosure, all of the patents, either on the molecule or in the medical imaging device. Myself and Dr. Wang were the inventors. So in the beginning, I'm like interested to show what the digestive cancer, and if you see this like left cartoon I'm showing here, like digestive cancer includes like esophagus, stomach, mouth, biliary, pancreas, colon, rectum, and intestine. Includes almost like about 80% of the overall human cancers. And most of the hollow organs, including esophagus, intestine, colon, rectum, etc., like they are endoscopically accessible, and the management of these cancer is usually routinely done by endoscopic devices. With the right, I'm showing this pie chart here. This is the overall incidence versus the mortality of the global cancer. So if you see here, India also kind of like shares the big, portion of the in terms of both incidence as well as in the mortality. So every year around like 18 million new cases are diagnosed with different types of cancer, and 9.6 million deaths. And over this, like around like 80% of the cancer covers like related with the gastrointestinal related cancers. So my previous like boss, he was like a physician scientist, and he was gastroenterologist. His interest was kind of like making the bridge between the existing technologies and how the molecular imaging can help. So the practice like throughout the world is like for the diagnosis of the cancer includes like different white light based endoscopic imaging, which includes like narrowband imaging. These are the patient like endoscopic images from the colon cancer. As you can see here in the narrowband imaging, which utilize the different filters, the combination of green and blue light, and it measures the absorbance from the hemoglobin, which also absorbs light around like 440 to 500 nanometer. And this changes the change the color because of the absorbance of the hemoglobin in the diseased area as compared to the normal healthy tissue, and you can kind of distinct the disease. Also, another device is chromoendoscopy, where you inject some of the non-specific contrast agents such as like indigo carmine dye that change the tissue architecture in the tissue and separate the diseased tissue compared to the healthy tissues. Another common technique is autofluorescence. So this utilizes the endogenous fluorophores from the tissue, such as collagen, and there are a lot of endogenous absorbing materials in the tissue, and with the and they absorb the light and emit in a different wavelength, and with the light you can detect the emission in the different color. While these all are great tool for the early stage of the diagnosis, but with the growing biologics and peptide-based drug or the small molecular-based drug for the different disease, molecular imaging is emerging tool to know the complex biology because the cancer is very heterogeneous, as well as to monitor the therapeutic efficacy from these drugs in the early stage. So this cartoon shows molecular imaging is like multidisciplinary component. As you can see here, like you need like the expertise from different area. It starts from the biomarker discovery from the tissue, then you design the molecular targets. Then there are several imaging modalities, includes optical imaging, PET, SPECT, Raman, MRI, ultrasound. Then to do the molecular chemistry is another component. Each modality requires a different molecule for the readout, and to design those molecules, someone needs with the chemistry experience. Then, based on the modality, those molecules need to be labeled with the in case of the optics, it will be with the fluorophores. For the nuclear imaging, it should be with the radioisotope, etc. Then once you have this molecule, then you need to validate again in the molecular level, cellular level. Then in the animal models, then need to be done the pharmacology, toxicology in the large animal models. Then with the approval from the regulatory authorities, then you can translate these imaging agents into the patient. I'm going to show how the molecular changes in different types of cancer. This is the example in the colon, and how we can target in the early stage using these pathways and identify the biomarker. So, for example, if you can see here in the left, this is shown here in the colon, including the upper colon and the distal colon. And when the disease starts actually in the mucosal epithelium, it starts as a kind of small bump you can see here. With the progression over the time, these bumps can be bigger, and after progressing to the cancer, it metastasize and transfer to other organs such as liver, lung, etc. And within this progression, the molecular pathways are different, and those molecular pathways can reveal the different biology which help us to identify the biomarker. So this is the molecular pathogenesis that happens in the colon cancer. I'm showing here the pathogenesis occur in the left colon, which is this distal colon, as well as in the right colon. So the molecular pathways are slightly different. And this cartoon was first published from one of my co-mentor, Professor Eric Fieron, who is now chair of the Comprehensive Cancer Center and professor at University of Michigan, and known as Fieron-Bogelstein pathway diagram. So when the mucosal healthy tissue start progressing to the cancer, you can see different genetic or the molecular events. For example, in case of the left side colon cancer, like mutation in the APC, mutation in the KRAS, loss of P53, etc. Similarly, in the right colon, like these pathways are little bit different. You see like more microsatellite instability and the different genetic mutation, including MLH1, MSH2. So these genetic events can give the window for identifying biomarker. Then you can design the molecule against those biomarker and use those molecules to detect these lesions in the early stage. So this cartoon shows the molecular events change in the Barrett's esophagus. Barrett's esophagus is another like digestive related cancer. And this is like compared to colon cancer or some of the other GI related cancer is more heterogeneous and more genetic events occur in the early stage. And the current practice, at least in the US, is whoever goes for the endoscopic practice in the clinic, if they see the Barrett's esophagus, that need to be further follow-up every year to see whether the disease is progressing or not. So this cartoon is showing how the endoscopic image looks like in the Barrett's esophagus. You can see this like patchy salmon color, like pink salmon color mucosal layer within the esophagus, and it's hard to find where the disease is basically using the white light imaging strategies. The surface of the epithelium looks very similar. And this varies also like based on the expertise of the endoscopist. There are some recently published paper and the detection sensitivity or the success of the detection relies on the expertise of the endoscopist. So our goal was if we can identify any biomarker that can target in the early stage cancer, for example, in this case, high-grade dysplasia or the low-grade dysplasia. So these are the events that occur before progressing to the cancer, and use those molecules and use the fluorescence device to detect these lesions. So I'm going to show some of the clinical data, what we have translated from the bench work to the clinic from these two cancers today. So first of all, as I have shown before in the multi-component molecular imaging pie chart, so you need to identify the target. So this is the work that was done from the Barrett's esophagus tissue using the RNA sequencing. As you can see here in this heat map, the green is are the genetic changes in the healthy tissues, but whatever you are seeing in the red color in this heat map, these are the distinct genetic events that occur in the advanced stage cancer as well as high-grade dysplasia. So our goal was like to see what these like these genes are and what they express in the surface as compared to the normal tissue, and use this information to design the molecule. For example, here you can see HER2, EGFR, like common kinase proteins that are overexpressed in the high-grade dysplasia or in the adenocarcinoma. So once you have this information, then our goal is, because molecular imaging, especially using the optics, light cannot penetrate the too much deep, and you need to have the kind of cell surface target, and we validate those findings using the immunohistochemistry or immunofluorescence. I'm showing here the immunohistochemistry of the HER2, which is one of the common marker that occurs like throughout different cancer, and especially in the breast cancer. And if you have like HER2 staining positive, then doctors refer those patients for Herceptin because there is already drug for that target. So, and these targets actually overexpress well in other types of cancer also, for example, in esophageal cancer as well as colorectal cancer. And we choose like this as an example to show how we can design the molecule. And as I mentioned before, to do the molecular imaging, making the molecular probe, which requires the chemistry expertise, is very important. And there are like multiple modalities, that probe modalities that can be used throughout these imaging platform, which includes the activatable probe, where you have like the molecule which have like two different fluorophores. Either you can excite using the FRET mechanism, or some molecules like they go inside the cells, then activated by protein, cleave and release the fluorescence. So the mechanism is doing the readout by the fluorescence. Then the huge market, basically in the antibody-based biologics therapeutics, by 2025, in the industry have predicted the biologics market would be somewhere around like $456 billion, and people are working on antibody-based either imaging agents or the therapeutic agents, like everywhere in the pharmaceutical industry. Then small molecule, with the approval of some of the probe that are commonly used in the clinical practice for doing the PET or SPECT, for example, monitoring the glucose metabolism using 18-FDG is like common agent already approved in the clinic, and there are several other small molecules in the pathway in the pipeline from different companies who are looking for developing better PET imaging agents. So then peptide is another modality, and basically my previous lab we were interested more on the peptide. Because of their small size, they can be selected using the different types of screening approaches, minimal toxicity, and like lower cost for the production. As well as peptides, they can be spread in the during the endoscopic procedure and bind to the target rapidly compared to some of the other modalities, because antibody is big molecule and need more time to reach to the target. Then there are some other like oligo-based FMR or other things. So my work basically was focused on the peptide, and I'm going to show some of the work from this imaging probe modality, how we have developed these peptides, and how we use them to do the molecular imaging in the patients. So as I'm showing here, this is the cartoon that shows the distribution of light from the visible range to the infrared range. And optical imaging actually provides like very good window for doing the multiplex imaging, because the cancer is heterogeneous. One target cannot cover the overall cancer, and because of the heterogeneity, if you block one target, other target can activate. So multiplexing could help in terms of understanding the better biology, and in that case, like optical imaging is more suitable and easier to do compared to nuclear imaging. So for doing the optical imaging, as you can see here, I have put some of these fluorophore structures that excite in the different region. The visible or the UV window is okay to do the in vitro imaging, microscopic imaging, but really for doing the in vivo imaging, light in this range actually can be interfered by the hemoglobin that is present in the blood. The hemoglobin observance is very high, but if you go towards like near infrared or far red, the hemoglobin observance is minimum, where you cannot see much tissue autofluorescence from. So there are several dyes that you can use within this window, conjugate with your probe with the binding motif, and you can perform the imaging in the infrared range. Also, light penetrates little bit deeper compared to the other region in this range. So this is the ideal window for doing the in vivo molecular imaging. So as I mentioned before, this is the cartoon that shows how we have screened these peptides. Phase display is one of the common screening technology that is widely used in the industries for screening the biologics, including antibody, different peptides. So how this screening technology work? You have the phase expressed in the bacteriophage. So I'm showing here, this is, for example, is M13 bacteriophage, you can express the peptides on top of the, using the coat protein on top of the bacteriophage, and one copy of the phage can carry 4 to 5 copy of the peptide. Then you use this random library, which is like very diverse, around like 10 to the power 11 number, and use this library against any target, which is kind of considered as a substrate, you can call, and screen for multiple rounds. And over the enrichment process, during the multiple cycles, at the end, when you pick up the clone, you will see the enrichment. If those clones are specifically binding to your target, then you see the enrichment, for example, from several thousand clones to few hundred. Then you pick up those clones and do further validation to pick up your lead candidate. So this is kind of chemistry group, and I thought to show some chem-draw, how we make the peptides and what are the chemical steps. And once you have the clone from this phage screening, then we need to synthesize peptides, translating those base pairs to the amino acid. And this was done using the solid phase synthesis. And basically, like, peptide synthesis is automated in this world, and there are so many automatic synthesizers you can use to make the amide bond between two amino acids. So this is how the chemistry works. You have the amino acid, then activate that amino acid. So other end you have the primary amine. So with the loss of water, you can make the amide bond. So activation can be done using like different activating reagents, and the intermediates are different based on whatever activating reagent you use. That includes halide, active ester, mixed asymmetrical anhydride, etc. So using this technology, we have made like the target peptide as well as some other control peptide. This is just, I'm showing one example here. It was 7 amino acid long peptide. Then we put the linker in between the dye as well as the peptide to separate the dye so that it cannot interfere the binding motif. Then basic characteristic includes the SPLC and the mass spec. So then we did like different kinds of validation. The way we have screened this peptide against different cancer cell line. So these are the esophageal cancer cell line. As you can see here, the green color is coming from the peptide, and the blue is just the nuclear staining. And it binds well with the different esophageal cancer cell line, but didn't bind with the healthy cell line, which was derived from the Barrett's esophagus, not from the disease organ. Then we quantified this, and you can see here the maximum uptake on the cancer cell lines as compared to the other control cell line, as well as there was no binding from other control peptides. This was another validation done using the flow cytometry. Then before translating this peptide, we need to validate how this peptide behave or perform in the human tissue. So we were collecting like tissue from the patient, and this was one of the tumor tissue collected directly from the patient. We had the imaging lab inside the hospital, and as soon as we received this tissue, we stain this tissue with the peptide. And as you can see here, the white color is showing in the fluorescence, and compared to the other area of the tissue, it's just like glow in the cancer area or the advanced stage, which is high-grade dysplasia. So then the pathologist confirm whether this tissue is disease or not, and these are the histologic images. So these are the quantified images. You can see here the peptide specifically bind with the disease tissue as compared to the healthy tissue, which is squamous or the Barrett's here. Then we got the IND approval from the FDA to translate this peptide in the clinic, and we had the molecular imaging device, which was confocal laser endoscope. As you can see here, we were using this peptide by topical administration, then wait 5 minutes so that it can reach to the target. That's one advantage for the clinicians, that they don't have to wait for a long time if they use peptide as an imaging agent, because of the small molecular weight, they can reach to the target much faster. So just after 5 minutes, we washed the unbound peptide and started imaging. And these are the fluorescence images collected from the same patient. And you can see here, like, there was no binding or minimal binding in the squamous or the bearers, which are healthy tissue, but there was significant binding. And you can see this was the laser confocal microscope and cellular architecture within the diseased tissue. And then we confirmed this by histology, and the tissue specimen was submitted for pathology, and pathologists evaluated the disease, and they turned out the high-grade dysplasia as well as cancer. Then, as I mentioned before, we were interested in doing molecular imaging in different types of digestive organs. So that previous project was on the esophagus, and we did the similar strategy to screen the peptide for the colorectal cancer as well. So this is just the structure of the peptide. We have screened using the same technology against the colorectal cancer cell line. And we did the similar pathway for validating these peptides in vitro. And this was the clinical trial identifier, also enrolled in the NIS clinical trial website, clinical endoscope. And they designed this fluorescence endoscope for us, and which is like this same version, just have the fluorescence filter. And this is the standard endoscopic image collected from the colon. You can see here, like, this is the suspected disease area, showing in the like little bit bump as compared to the normal healthy tissue. And when we spray this peptide to see whether this peptide hooked to this region or not, then after spraying the peptide and a collected image using the fluorescence fluorescence endoscope, the peptide is specifically bound to the lesion, but not with the surrounding mucosa. And this is just the reflectance image to suspect the background, how that correlates with the fluorescence. And we published this in Gastro, one of the leading journal in the GI field, in 2018 as well as 2015. So then we were continuously like working in improving the binding or the specificity of the molecular imaging probes. As I mentioned, like we were kind of screening these peptides using the phase display. If you see the structure of the phase shown here, like it contains like 4 to 5 copies of the peptide. And the work that I have shown before was just using the monomeric peptide. So when you do this screening, like because of the avidity effect from is like, the phase has the higher binding affinity with their targets as compared to just using the monomer. Then I thought, like, if we can mimic the binding property of the phase, we need to design the peptide in a multimer form so that it can maintain the avidity and maintain the binding affinity similar to the phase molecule. So I used the common branch core at the C-terminal side of the peptide, and then anchored like all, one copy of the peptide, but 4 times in to make this multimer, just to look like similar to the phase molecule. So this is our hypothesis. If you have the multimer peptide, even though the target expression is low, so it has higher chances to bind to their target, even with the low expression. So this is just the structure of the peptide that I have designed, and one peptide within this core has 4 different peptides, and it was labeled with just one dye at the C-terminal side. So then we were working to validate this peptide with our collaborator, who was one of the leading name in the field, and he's right now the chairperson of the cancer center at University of Michigan. He's the one who developed or who identified the molecular pathogenesis pathway in the colorectal cancer. And he developed this mouse model that can mimic the disease from the human. In the human also the APC mutation occurs in about. So this is like little bit old the harvested colon from the mice, and you can see here like multiple polyps, which were like some of them have like pathology with the adenoma, which is early stage cancer, and some of these polyps has cancer. So this is how we used to do the endoscopy in the mice, as you can see here in this white light video displaying this polyp start growing, and we used to evaluate them whether there are polyps or not, and then use this mice model for most of the preclinical validation. So then I have used that multimer peptide as I saw before to see whether we can detect these lesions as early as possible. So we have used the young mice, like started from like 4 to 5 weeks, and in that case actually they have like very small size polyp, which are like hardly detectable using the white light endoscope, as you can see here. But when we use the fluorescence endoscope, you can see much better kind of glowing the tumor as compared to the healthy tissue, and the specificity was like pretty good. And we then harvested these tumors just to confirm whether there is disease or not, and pathologists confirmed that there was the early stage cancer, which is called adenoma by pathological word. Whereas like we also use monomer, one copy of the peptide, but there was not much binding in the small size tumor. Then we did like further validation using ex vivo, other like whole body imaging tool. This was just one harvested colon using the IBC imaging, and all of these polyps, like they were shown in the fluorescence as well. Then, as I mentioned, like cancer is very heterogeneous, and we were like very interested to solve the problem by doing the multiplexing. As you can see in this example, there are many cell surface targets that provides the opportunity to design the imaging probe that can target these biomarkers, and you can use those multiple biomarkers in a multiplex setting. And we were collaborating with Professor Eric Seibel at University of Washington, and he is the one like who designed this scanning fiber endoscope. Like currently, like used in the clinic for doing like different endoscopic procedure. So this is the just like optical design of this scanning fiber endoscope. It is like very thin flexible probe, which is like around 1 millimeter in size, that you can use easily for doing the small animal imaging in the colon or in the esophagus. And this endoscope has like 3 laser sources that can illuminate the light simultaneously, and you can collect the image basically like similar to the confocal microscopes for the ex vivo work in a large field of view to read the multiplexing from 3 different targets. And then we use this technology first to use in the mice. We use like 3 different peptides labeled with 3 different dyes, and you can see here pretty good specific accumulation or the homing of this peptide in the dysplastic lesion in this mice model. Then besides that, like we were also interested like by if we don't have the right instrument, scanning fiber endoscope is not like clinically approved, so you have to design the endoscope which can deliver the light to collect the image from 2 or 3 different channels. We were kind of interested like making the bispecific peptide, where you don't have to label the peptide with 2 different targets. So we designed this peptide that targets like 2 different molecules, which is EGFR or HER2, commonly expressed like kinase protein in different types of cancer. And to modulate the binding, we like made like huge library of the molecules with different linker length. We use like PEG linker to separate these peptides, and conjugated with the dye at the C terminus. Then use this peptide to target these two proteins in vivo. So again, we use the same pathway for doing the validation, and this was also validated in the model implanted with the one of the cell line, which is OE33, as well as SKBR3, which is breast cancer cell line. And as you can see here in the top panel, the peptide home to the tumor with higher contrast as compared to the control. And this is right now in the clinical trial, and this is the clinical trial identifier. Then one modality is not enough, so we are also interested in looking the specificity of this probe using other modalities. So as Dr. Pan also outlined in his talk about the photoacoustic imaging, which requires the acoustic waves and release, you can measure the photon from the sound. This has like some interesting feature, even though this is not whole body imaging, but it can penetrate much deeper as compared to the fluorescence based imaging. So as you can see here, like this is the white light image the tumor is showing with this dotted line, and we use the photoacoustic imaging, and our readout, actually we injected the peptide with the absorbance in the NIR range. You can just use rather than using the endogenous absorbance, we use the peptide, whether that peptide home to the tumor specifically or not. As you can see here with the circle area, that's the tumor, and we saw like almost like 3 millimeter depth of the penetration in the photoacoustic. Also, we confirm that whether the tumor is there or not using the ultrasound. Also, you can see here the black hole here is the tumor detected by ultrasound. Then we use we were kind of doing a lot of prototyping of other confocal based laser endoscope that can be used in a surgical tissue, or by doing the rodent survival surgery in the mice. And we use this this tool by opening the tumor and how the probes are localized. And as you can see here, these are the images collected by opening the tumor, and you see the specific accumulation of the probe in the tumor, but not in the normal area. As I mentioned, we were continuously interested in looking the specificity or expanding the potential of this molecular imaging approach. So we conjugated some of these probes to do the nuclear imaging. So this is the example for doing the SPECT imaging. And we designed the different linkers, and for doing the nuclear imaging, especially with the radioisotope, you need to do the chemistry in relatively short amount of time because of the half-life of the radioisotopes. So we designed this chelator by conjugating this bipyridinyl molecule. So these 3 nitrogen can provide the conjugation site for the technetium, and we did this labeling in the radio isotope making facility. And we use this technetium 99 meta-technetium labeled probe for doing the SPECT imaging in the mice colon. As you can see here, the uptake was huge in the bladder, but there was tumor detection as well. But over the time, there was minimal signal in the bladder and some signal retained still in the tumor. Then we were kind of interested in looking the further potential of these peptides, whether we can use these peptides for targeted therapy or not. So rapamycin is one of the common small molecule drug for the mTOR pathway, which is pathway in multiple cancer, including colorectal cancer. And if you use the, chemotherapy has some potential, as you all know, that it has huge potential, but it has several side effects also. And the outcome is dependent on different patients. Some people get more side effects, some people get less. So we were kind of looking whether we can minimize the side effect by combining this molecule in the nanomicelle type of formulation using this targeted peptide. And these are the again like images from the same mice model, as you can see here, compared to the just like PBS vehicle, the tumor size was reduced over the time. The treatment started at day 0, and in about like 32 days, the tumor got shrink. And we found that like less toxicity compared to the rapamycin with the peptide-based formulation. So to summarize my talk, I would like to say that molecular imaging is multidisciplinary field in the intersection of multiple discipline, and chemistry is like key component in that system. Also, molecular imaging is part of the translational research and may be applied for biomarker-driven personalized therapy. Key components of molecular imaging are use of imaging modalities with different performances, development of probes, ligands, detectable in vivo. Molecular probes, including peptides, can illuminate the tumor for guiding biopsy, surgical resection, as well as monitoring therapy. So with that, I would like to kind of acknowledge my my mentor, Dr. Wang, who is here, and multiple collaborators, other faculty members from University of Michigan Health System, many lab members. I have worked with like many clinicians to do the clinical trial on these molecules. So I would like to thank them, as well as I have worked with regulatory teams to file the IND application on these molecules with the FDA, as well as pathology team and GI pathologist. Worked with like multiple industrial partners, either to make the molecule, GMP grade molecule, or make the molecular device. And without funding, like this work can't be done. So most of this funding was from the National Cancer Institute, NCI, and I would like to thank everyone. And thank you everyone for listening me. And if you have any question, I would be happy to questions.
Thank you, Dr. Joshi, for the very nice, lucid, and illuminating lecture. Now the seminar is open. This can be conducted by to conduct this session.
Thank you, Dr. Joshi, for your excellent presentation. And there are many questions. Out of that, very few will be taken due to time limit. So, is this probe specific for cancerous cell? The probe?
Yes. So we have done this, early stage screening in an unbiased manner using the cancerous cells, as I have shown for the esophagus, like those peptides were screened against the H460, which is esophageal cancer cell line. And later on, like we were interested in, making this probe more specific for certain targets which are overexpressed throughout the different disease. For example, HER2, EGFR. So in that case, like we have screened those peptides against the cell surface proteins.
Okay. So what is the cause of colon cancer at the early stage, age 25 to 30 years of human life?
So that's like kind of like very good question, and this depends on the family history, genetic mutation, lifestyle. Colon cancer was usually like more common in the Western world, but these days, like if you see the epidemiological data, this disease is like increasing in the Asian countries or the developed countries, because they are trying to adopt the different lifestyle. So usually like after 50 the chances are high. If there is no family history or something like that, chances are less to see the colon cancer before 50. So familial adenomatous polyposis path is one which is kind of like coming based on the family history or the genetic mutation in the family. That depends on just like the family history. I can't comment on the kind of exact statistics in that age range, but the chances are high after 50. That's how in US or in Japan or some other countries like they do the practice.
Yes. Thank you, Joshi. So now I request Dr. Anjay Maji to ask few questions from YouTube chat box.
Thank you, Dr. Palani, and thank you to Dr. Joshi. Thank you for your excellent presentation. So I have few questions from YouTube chat. So Bidisha Bora asking, does lipophilicity and hydrophilicity of the molecule matter for designing it for cancer imaging?
Yes, that's like great question. The hydrophilicity is these days like many companies are working on the cell penetrating peptides. If you remember any sequence for the cell penetrating peptides, for example arginine 8 or multiple arginine containing peptides, they are basically like very hydrophilic. So if the molecule is hydrophilic, it tends to penetrate and carry the cargo inside the cell. But these molecules are not designed for the penetration purpose, because we were only interested on the cell surface marker, and imaging cannot go deep to image the intracellular target. So definitely, yeah, that's like more kind of decision making factor. We are working with the therapeutic molecules to deliver the drug inside the cells.
Thank you. Another question. Sudhir Gorai, he is asking how the selectivity is obtained with your bioimaging dye bound peptide for the said GI cancer cell over non-cancer cell.
So we do the screening when we screen these peptides or the lipase library. We do the non-specific screening against the normal cancer cell line also to reduce the non-specific binding from the clones, and then we validate through our different assays using different cancer cell line, using different proteins, using different tissues just to confirm the specificity. And non-specific, minimize the non-specific binding from the healthy tissues before translating to the patient. Yeah, there was a robust pathway to validate and confirm the specificity.
Thank you so much, Dr. Joshi. Now I'll transfer this call to Dr. Aditi Bose.
Thank you, Dr. Maji. First of all, I would like to thank our speaker, Dr. Joshi, for his very interesting presentation on molecular imaging and specifically the use of peptide for targeted therapy. Thank you very much, Dr. Joshi, for being with us and enlightening us. And I would also like to thank Professor Bijon Dash for chairing the session. And now we will move on to the last technical session of today's evening. And I would like to invite Dr. Arnab Haldar from President's University to chair this session and introduce the speaker to the audience. Over to you, Dr. Haldar.
Thank you so much.
Thank you, Aditi. Thank you, Aditi. Am I audible?
Yeah.
Am I audible to all?
Yes, you're audible. You're audible.
Thank you. Actually, this is the last session of this seminar. Chemistry is a subject of matter, and it is very much involved to improve the quality of life. Pharmaceutical industry and chemical industry both are very much dependent on the knowledge of basic chemistry. In most of the developed countries, research is governed by the industrial needs. So, proper understanding of the interrelationship between chemical research and industrial parameters is essential. The next lecture will be re-enriched by Dr. Pranab Haldar, who has vast experience in industry, project management, and research. His blending knowledge will help us to know the several ideas about industries, research, self-management, and personal development. Dr. Haldar is the head strategic sourcing, Sandros, Novartis, India. He received the Young Scientist Award from the Indian Chemical Society in the year of 2002 and enlisted in Marquez, New Jersey, United States of America, who's who in the world for the year 2010. So now we'll learn many things, so many things about the industry and research and everything from Dr. Haldar. Dr. Haldar, please.
Dr. Haldar, you are muted. Dr. Haldar, you are muted. Dr. Pranab Haldar, you are muted.
Dr. Haldar.
Yeah. Good evening, everyone. It's a great pleasure and opportunity to present in such an auspicious day in front of my two teacher. I can call it one of the trained philosopher guide, Professor Ray, as well as Dr. Kaur. Because of him, I am here today. So I can remember, I think 2006, 14 years before I presented my PhD dissertation in front of Dr. Ray. And though I speak a lot of industrial conferences, but today is my second presentation in front of Dr. Ray and other communities. So first of all, I just like to tell that after such a scintillating presentation from Dr. Pan and Dr. Joshi, which is like a starter and main course after the dinner. So it's just a dessert, and I hope that you all enjoy the dessert and flavor of the industry, how the medical innovations actually coming into the practical aspect. So with this few words, I just tried to minimize my talk in very short way so that and try to cover up more questions because it is related to the practical aspect of the medical development and the pharmaceutical industry. Just give me. Will you able to see my presentations? Yeah. So my presentation basically I will start to talk about something about the today's healthcare systems, then how India is in the world healthcare systems. Then I will go into the touch upon that how molecules will go to the medicines levels. Little bit brief on the drug development cycles where actually we can understand how the industry, academia, and research institute interaction is very, very important. And at last, I to cover how important is the career of a chemistry students and what are the pathways is there in present state of industry. So what does health means in present aspect? Is it health is only the body? It is not like that. So if you see our present situation in COVID situation suddenly, so we all are going through a lot of mental stress. It is not only the physical part, it is the mental stress. And another thing is the social part. You see the lot of cases, the asymptomatic patients does not want to go for the testing. Why they don't want to go for the testing? Because of social hideness. So health is just a state of complete physical, mental, and social well-being. And all the medical and pharma industry now today working on that part. It is not only the cure of the disease or infirmity. It is a person's happiness, a adaptability with the situations, and a balance with the environment. So our healthcare system basically working, and it is overall all the healthcare system is working. Now, what is the shift happened in last couple of years? Basic shift is happening. Previously, we are looking that how to cure our disease, because after curing the disease, we can overcome the illness. But is it important? Like Dr. Pan is telling that before COVID, we can test. Can we get some clue that at starting point, can we understand that COVID is there or not? So that's why it is very, very important, which we are listening from all above. Preventing is better than cure. So all the pharmaceutical industry and our healthcare systems moving towards the prevention, which is the wellness part of the healthcare systems. So now, basically, we are looking to the preventing of the disease, which is the well-being of our physical, mental, and social wellness. So what are the shift? In nutshell, we are moving from I to we, means illness to wellness. So how I to we? Here actually illness is with us, our mindsets. But wellness is for the society, wellness for the families. And now we are moving from the, already I told, curing to the preventions. And another good part is that nowadays, if you see that all the pharmaceutical companies are interacting with each other, it is they are not working in isolation. So basically, the companies understand that if we work together, we can resolve the problems very easily. And you can see Dr. Pan and Dr. Joshi also emphasize on the collaborations part. So medical healthcare systems and pharmaceutical companies also collaborating with each other. And last part is the digitizations. Not only now the medicine system, healthcare system is doctor, physician, and patients. Not like that. So what is happening nowadays? Actually, we are getting alert in the mobile digital area. We are getting alert that when you have to take what medications. If your medication is completed the course, you will get the alert for the next sessions. So this is the digital era. Now this is the world brief of the how world healthcare is going on. So what is India's contribution presently in this world healthcare? So India is not only contributing in the pharma sector. If you see, India is a great place for the medical tourism and alternate medicines and doctor and nurses. So I'll just give the brief of the some positive things. We always listen that Western countries are doing great in the healthcare systems. But actually, if you look at the data, if you see around 100 countries which covered around 80% of the world populations, the essential medications, vaccines, and medical equipment are supplied from India. The fifth generic drug of the world comes from India. And HIV, every third HIV drug comes from India. So you can see easily that how the India is contributing to the world health. And 70 to 90% of the essential medicine of the developing countries also India is providing. And if you see the data, whatever UNICEF is procuring the medicines from the underdeveloped countries, they are procuring around 50% from the India. So, and I am proud that out of this 80 to 100% whatever medical healthcare systems we are providing medicines, only Telangana and Andhra Pradesh region is providing 40% of our total medical requirements. So if you see actually this southern part of the India providing 40%, out of this only Telangana and Andhra Pradesh providing 40% of our medical requirement. And what are the benefit actually we are getting in India? If you see all the bigger pharmaceutical companies now coming in India, Novartis, GSK, Eli Lilly, all are coming to India. All are innovator company. Already India has their own generics company like Dr. Reddy's, Sun Pharma, Lupin, Cipla, who are working in and out for the medical healthcare systems. But nowadays, innovator companies also started coming in India. So what advantage are they getting? Actually, if you see the cost of the clinical trial in US or Europe, India's cost of the clinical trial is just 20%. And 1/6 of the doctors in US actually Indian origins. And if you see the Indian talent pool, which is very, very important, every year 7 million scientists or engineering graduates are passing in India, and 1,500 PhDs are qualifying annually in India. So you can see that the resource of India and innovations is growing day by day. And another important things for the innovator companies that Indian populations and the wide spectrum of disease condition helping the innovator companies to perform their clinical trials. And same thing, you can see that COVID-19 vaccine when we are other companies are developing, but actually we are tying up, Oxford is tying up of the Serum Institute and Bharat Biotech working with ICMR. So these are the benefits of the Indian healthcare systems, and we should feel proud that we are part of India and India is contributing this way to the world healthcare systems. But is it the end? So if everything is we are getting and people lifestyle is increasing, if you see the mortality rate also reduced, are we still not lagging anything? Yes. If you see the last year data, around 2 billion of people around the world are not getting necessary medications. And most important part, what is coming out, any innovator medicines which is coming out at starting, the cost is so high that million of people are going to the poverty line just to manage their healthcare systems. And millions are waiting to manage their disease. So I would like to add here one point, the word manage the disease. Because now everything is a management. It is not that disease happened and we are taking medicines. It is called now disease management. And it is part of the wellness time. So if you see that, we know that what we have to start our day and what we act so that this disease will not come. And because of this, all these reasons, today still pharma sector is growing high. And pharmacy sector is an exciting area for the career opportunity for the students. So after the Indians healthcare systems, I think we all are feeling proud for our Indian healthcare systems. I just thought to give a brief of the how the drug development is going on. So if you see, it is around a 13 to 14 years overall project when a drug is get approved, new drug get approved by FDA. And more important thing is that at first around 10,000 compounds are selected. From that, one compound get approved. And if you see the timeline at the initial stage, just for the discovery to preclinical stage, it will take around 6 to 7 years. And clinical phase, which is phase 1, phase 2, phase 3, and ultimately we are coming to phase 4, where actually regulatory submission, it took around 7 years another, and ultimately approval will also take 1 to 2 years. So this is the reasons actually any innovator medicines when coming to the first in the market, the price is very high because as per US law or Canadian law, after the publications of the first drug patent, you will get only 20 years opportunity for the market, and after that generic will enter. But automatically 14 years is over to develop a drug. So within this 6 or 5 years, innovator always try to get the profit and whatever they invested in the development. So that's why innovator companies' price is always high. And here we are lucky that India is full of the generic pharma health sector is there. And in the world, out of all generic pharma health sector, India is providing 55% generic medications. So with this inspiring story, let us move that. I would like to cover that what are the academic divisions is involved just from the selection of the molecule to the medications part. So starting from the finding the right approach, then screening, then modulations, modeling, then the optimizations, then understanding the effect, then API development, API synthesis, then formulations. After that, we go for the trials, phase 1, phase 2, phase 3, which actually we check the tolerability of the drug, then efficacy. And then we go for the pharmacokinetics and then regulatory efforts. So we see that all these cases, there is a great development of interdisciplinary subjects. Structural biology, computational chemistry, organic chemistry, medicinal chemistry, pharmacology, toxicology, regulatory affairs, drug discovery, everything is important. But when all these things are important, then how a healthcare or pharmaceutics can develop a product? So basically they work on the PPT systems. PPT is, we usually depend on 3 parts. One is the people, process and tools. And here important part is the peoples. Process and tools we can develop, but peoples is very, very important. So I just thought to share the thought. You see, today in Teachers' Day, we are wishing all the teachers whom they taught me or whom we go through that. But are we anytime telling that saluting our Google teachers? No. But if you see nowadays any information if you want, you can type in Google and you can get it. But we are still we don't remember or we never feeling any appreciation for the Google teachers. Why it is so? Because it is very, very important, our own thing, our own innovation, our thinking process. So all the medical companies, anything you want to learn, you can type in Google. Then what is the differentiating factor? Factor is the people. And here people is our young generations. So it is very, very important if you have to sustain and if you in medical healthcare systems, we have to grow our young generations and we have to groom them in such a way that they should feel passion from the starting from the college level onwards so that they can feel to contribute to the society in whatever the way, in academic and research or pharma industry. If we just go through the last year actually 2019 is very important year in the chemistry sections. It was the 100th year of IUPAC, in International Union of Pure and Applied Chemistry, 100th year celebrations, and 150th year of Mendeleev periodic table first publications. And there IUPAC actually disclosed that what are the 10 key chemical innovations which can change the world. So this is for our young generations. You can see that last just 2019, out of all the key innovations, I am just marked that green color, which is enhances selective organocatalysis, flow chemistry, and mechanochemistry. Already we are started applying in the pharmaceutical sector. So you see 2019, this came as a world-changing factor and already pharma industry started using this. It is very, very important for us to groom ourselves, not only depend on the theory, but to have to be habituated with our practical understandings. Let me give some examples. Enantioselective organocatalysis. We can remember that Professor Kesey in Harvard University invented one molecule that called eribulin mesylate, which is used for the breast cancer. This molecule has 19 chiral center, and Professor Kesey has developed this molecule ultimately in a NMR tubes. But today, pharmaceutical industry, we are selling these molecules and generating in a gram scale in the pharmaceutical industry. It is a 81 stage synthetic process, but still from the academic NMR tube, now we are in the plant and we are distributing these medicines to the breast cancer patients. So that's why it is very, very important from the how we move from theory to the practical aspects. And that's why it is important that we should understand and we should collaborate. Academic industry collaboration is very, very important. I'm coming to the flow chemistry. I think that Dr. Joshi was mentioning the peptide synthesis, amino acids. Usually people do in a solid phase synthesizer. But nowadays, we have a microwave assisted flow chemistry systems where you can put all the amino acids together, and with microwave systems, you can get directly the peptides. So this is the innovations, and it is well in India also we are using this microwave assisted chemistry in plant scales, where you will not get any impurity of other amino acids. And last part is the recursive extrusions, where actually pharmaceutical company working towards solvent-free process. We all know the green chemistry aspect, so this is one of the green chemistry aspects. I feel that with these three innovations, which is actually there are 10 innovations, actually very, very important for our young generations and influence them to come out from the theory, move towards the practical aspects of the life. So there are a lot of opportunities out there. I will just go through one by one. That for a chemistry students, is there any opportunity other than the academics? Yes, other than the academics, lot of research opportunities are there, lot of industry is there. Let me take my example first. So I was a synthetic organic chemist. I studied masters in chemistry, and then I did PhD in chemistry. Then I moved to the postdoctorate, did medicinal chemistry. But after joining industry, I started working synthesizing the molecule, which is called active pharmaceutical ingredient API. When I was working in this API, we realized that ultimately these molecules should go to the patients. That is the major benefit. I am synthesizing this molecule in the lab, but if it will not go to the patient, there is no use. That's why I moved my career. I felt that it is better to learn what is the other part of the pharmaceutical industry. I moved to the API synthesis to the management part, which is project management. It is not like technology management. It is overall a project, a molecule synthesis management. Later, I moved to the program management. Program management is a program which is called like a medicines where like COVID-19 is a program. Pharmaceutical industry is working on that. Globally, different companies are working. So overall program management. And now I move to the next curriculum, which is actually global strategic sourcing. So you can see learning from chemistry, you have a lot of options. Not only you have to stay back only in the synthetic part. You can move to the management part, you can move to the sourcing part, you can go to the quality part, regulatory part, and you can serve the societies. So with these few words, I can tell that so only the chemistry is important. Nowadays, pharma companies is not only working in the chemistry. So what is very, very important? Important is the cross-functional interface. And that's why industry is very, very important. The difference between academia to industry is that if you go to the industry, you can learn that how cross-functional interfaces is happening. It is not only chemistry and biology or the nanoscience. It is the chemical engineering. It is the artificial intelligence. All we are working. Even statistics. I think we all know the quality by designs. So when a molecule, a pharma company is developing, so statistician get involved. So we are statistics, artificial intelligence, chemical engineering, pharmaceutical pharmacophilia, organic chemist, inorganic chemist, analytical chemist, and influence a drug product development. So it is very, very important. If you go to the only to the industry, you will understand how cross-functional interfaces can happen. The most important thing, it can teach one of the important part of our life, which is accountability. So that's why a lot of the management talk, we call that we should not talk I, we should talk we. So from where this accountability, I to we? So it will come from the industry. If we go to the industry, when you synthesize a molecule, any research or academic industry usually synthesize a molecule in the milligram or few gram scales. But when you synthesize a molecule in the industry, there are three factors always you have to keep in mind before synthesizing molecules. The safety factor, the financial factors, and the timing is very, very important, because none of the pharma industry is here just to free contributions to the society. It is not like that. They want to serve the society, but along with the profit also. So obviously it is very, very important. We have to take care about the cost and the timing, how quickly we can develop this molecule and we can market these molecules. Another part is the intellectual property. So if you go to the industry, you can understand basically in the generic industry, when you synthesize a molecule, we can get lot of constraint from the intellectual part. The same molecules, which is innovator molecules, is protected by IP rights, but a generic company try to develop a different process which will helpful to understand the intellectual property of the molecules. Last but not the least, it will help us to increase our business as well as communication skills. It is very, very important in the present society. It is not only should be a good human being, it is important that we should create a good society. And that's why it is very, very important we have to create a culture of trust. And this trust will come from our business skills and communication skills. Now for the students, I just thought to put some slide that we are talking that theory to practicals. So what is important for our young generations who are still pursuing, maybe studying or pursuing research? So you see lot of thought process, lot of chemistry, chemical reaction, mechanisms, theory we are reading. But ultimate important is that when we are doing the practical experience, it is very, very important that we should be mindful enough. Why it is important to be mindful? When we will be mindful, then we can concentrate more on the reactions part and we can understand and get the information more. Now there are different procedures or wellness programs are there how to be mindful. But I just thought to share that you can easily do the meditations part for our own mindfulness. And it is very, very important for all of us to practice the mindfulness. What is mindfulness? Mindfulness means being now here. Means what I am talking about today, I am totally focusing and concentrating to whom the audience I am talking. So like that, when we are doing some experiment in the lab, we should understand our focus should be only to the experimentation part, not the other things, WhatsApp message or some what India is scoring like that. So it is very, very important we should practice mindfulness, which will help us to increase our awareness, acceptance, and so now lot of cases people, so I just tried to capture few things which actually I received lot of questions from the students and teachers before I take up the questions. So they told that when I go through the study, it is I think around 14 to 16 year study, then PhD, then we are puzzled, we'll go to the industry, we'll go to the academic job, we'll go to the research, what we will do. And now present situation, COVID-19, it is one of the stress factor everyone is facing. So what to do? So it is very, very important. If we cannot change others, we should change ourselves. So let's try to change ourselves. So what to do? The basic simple tricks for all of us, how to change ourselves. So this change will come from ourselves is within. So what is? It is our awareness systems. We have to understand everything, who we are, what is the purpose of life. So we have to think about self, people, and the present situations. We have to increase our awareness capability. And decision making, it is very, very important. Not only that, when we go from the school to the college, which college we study, which topic we take. Second part is that, okay, after that, shall we go to the research or shall we go to the joint entrance engineering and medical? Then the again question will come, shall I study organic chemistry, inorganic chemistry, or analytical chemistry? So these cases, decision making is very, very important. And the basic tool of the decision making we can practice that is called whole body decision scan. So if any time this decision making thought process will come, we can just sit quietly and close our eyes and we can scan, means we can start thinking our body starting from brain, heart to the gut. And when you think one of the topic, the case when you feel the uneasiness, you will understand that topic is not for you. This is one of the simplest way you can easily take the decisions what you can do in your life. And as I told, clear-cut communication is very, very important. Last but not the least is very, very important, the adaptability. Adaptability is one type of agile. So you see that we never thought that we'll present or interact in a digital world. Usually we go for a presentations and conference. This is my 5th presentation in a webinar. But it started only from the month of May. Previously, I never thought I will present. So this is now, even we are practicing how to present in a webinar, because it is very easy for me to present in front of audience when I am face to face. But now I am practicing myself how to present in a webinar just sitting in a chair. So this is called the adaptability and agility. And it is very, very important for the students also. We should be agile and adaptable with our society and present situations. Whatever the situations come, we should be capable enough to handle those situations. So at the end of my things, I tell that it is not the end. It is the journey. We are starting the journey. And it is very, very important. We should not just going through the chemistry. Going through the chemistry means just to reading the theory or textbook and performing the whatever the experiment suggested. It is very, very important now. We should grow through the chemistry. Grow through the chemistry means we have to understand the practical aspect. We have to understand the what world wants from you. It's very, very important how the healthcare system is progressing well. And another, but last but not the least, you have to question ourselves. What is the purpose of ourselves in this world? If we know what is the purpose, then obviously we can easily question our answers that what we want to do. So it is very, very important. Rather than going through the chemistry, let us go through the chemistry. With this few words, I just thought to close my talk, and I am very open to accept any questions.
Okay. Thank you, Dr. Hallar, for your wonderful lecture. Now, this is the time for the question session. Actually, the question session will be coordinated by Dr. P. Shashi Kumar in Google Meet and Dr. Anjay Maji in YouTube. First of all, P. Shashi Kumar. Shashi, please.
Yeah, thank you, Arnab. So, first of all, thank you for very, very nice lecture. And I'll ask only few questions because of time limit again. Has any original generic drug been developed in India recently and made to marketing? So this is the first question. And regarding this, they extended such a manner, pharmaceutical industry in India preparing only the formulation developed elsewhere.
Yeah. So let me answer first thing that yes, India actually all Indian companies are generics company. Till now there is no innovative molecule came from India. So whatever the molecule is coming from India, it is the generic molecule, and it is the original generic molecule. Generic cannot be like original or something. Yes, it is a generic molecule. And blockbuster drug which you can use every day, you can understand that omeprazoles, pandomeprazoles, all blockbuster drugs for the generic molecules. And presently I can see, you can understand that here all the favipiravir, remdesivir, which is actually coming for the COVID-19. All cases, few cases, Indian pharmaceutical companies are working together to develop this drug. Otherwise, like Dr. Reddy's has tied up with Fujifilm, who is the innovator of favipiravir. So this favipiravir drugs actually generic versions Dr. Reddy's is bringing into the market, and other companies already brought into the market. But those are the tie-ups of the companies already we have, and all Indian companies, whatever drug you can see in the market, all are generic drugs.
Okay. So next question is that it is only the human genius which can come out of the limited and representative thought based on old memory and enter into the source or realm of infinity knowledge of discover novel things. Why the scientists are more prone to follow old time bound methodology to discover COVID-19 vaccine? Why there is no novel way without following the old methodology? Is this the lack of understanding which the scientist community is suffering from?
So I would like to take it up that scientist community is never lacking with any novel thought process or something. That's why scientific community still exists. So that's why I told that we are always thinking a novel thing. Otherwise, anybody can search in Google and you can develop a drug. It's not like that. It's that we are always thinking, and all scientific community is thinking. I showed you that one novel drug if come into the market, it takes minimum 13 to 14 years just to bring a one single drug into the market. In COVID-19 situation, it is just, I can see that 6 to 9 months case. What scientists prefer here to work in the two aspects. One is the vaccines, another is the drug. So easiest approach, if you want to reduce this 13 to 14 years to a month, what is the easiest approach? Approach is that to understand what is the molecules, and we can understand this is one type of antiviral type of drugs. So that's why you see all the drugs which is coming into the market, like favipiravir, remdesivir, all these drugs are already old drugs for the innovators, but it is coming for the generic versions to fight with the COVID-19. It is nothing like that. Scientists are thinking, yes, scientists are thinking and scientists are working on that. And as I explained, it is not only one scientist is working. All the world modeling, statistics, engineering, simulations, everything is working well. And I am sure very soon we can hear that novel vaccine is coming, as it is easy for us, but a novel drug will take time. Because simply if you see that clinical trial approach, phase 1, phase 2, phase 3, and ultimately to phase 4, earliest time it will take minimum 6 years. And if we see the latest things, even in the vaccines, if we see actually the last vaccine which came in the world easiest, means what I can say, it took 7 years time to come into the market. So that is the earliest vaccines came into the world. But still now in COVID-19 case, we are talking in a 7 months that should come. So my request is that just keep the patience and collaborate with each other and keep the respect to the scientific world who are day and night working to bring out the medicines and serving to the patients.
Thank you, Dr. Halder. Now, I request Dr. Anjay Maji to ask a few questions from YouTube. Dr. Maji?
Hello, am I audible?
Yes.
Yeah. Thank you, Dr. Palani and Dr. Halder for your nice speech. So, I have a question that Anushka Chakravarti asking. She is from Jadavpur University, and she is asking, can you suggest from when we should think about pursuing a management degree, after a bachelor's degree or a master degree?
So let me answer it in a different way. Actually, management degree we can pursue from your primary career. So when we are at home, you can learn your best management degree from your mother. How your mother managing the home. So you don't require a practical management degree, just a time that after this career we can learn management. You can learn management from your mother very easily. It is just like short way I am just answering. Yes, but to answer your questions, if you learn the project management, I can only answer one thing. You watch Kung Fu Panda. I think it is very, very important. We all watch the Kung Fu Panda cartoons. You watch Kung Fu Panda, you can understand all management lessons. And if you want to go to the management leaders, just read Mahabharata. Krishna was the best project manager, project management leader till in the world. If you understand Mahabharata and Lord Krishna, you don't need to do anything or read any management book. Okay. Yes. Now, last to your questions. So it is always, that's what I told. It is very, very important. We have to understand what is the purpose of our life. And when you understand this purpose, it is very, very important. We have to know what is our passion. So like me, I am in a management world now, but my passion was synthetic organic chemistry. So it is very, very important. If you understand what is your passion, nothing will stop you, or there is any time or specific career path you can tell to understand, go to the management or academic. Any time you can learn management, and management, in my words, it is more practical aspect. So I would request you in your BSc, MSc or PhD, please concentrate more on the practical aspects of the whatever you are doing. And once you move to the world, you can easily understand the management in more practical aspects. But obviously, degree is very, very important. So, long story short, start learning management from your mother. Read Mahabharata, watch Kung Fu Panda or Shri Krishna, whatever the serials, you will learn management 70%. And later, after your PhD or MSc, when you go to the industry or outside world, obviously you will get practical knowledge.
Thank you very much, Dr. Halder. Another question, and this question asked by several candidates. So, what is the career opportunity after doing PhD in chemistry? If you can elaborate a little bit.
Yeah, obviously. So after doing chemistry, so you can go anywhere in the pharmacy. I hope this case question is for the PhD in chemistry, not the other. Considering that it is PhD in chemistry, you can go to the, I think I have shown in the slide also. You can go to the, first thing is the any pharmaceutical industry, chemical industry, agrochemical industry. You can go to the academic research. You can go to the academic professions. But nowadays it is very, very important. Even if you see the actually, we can tell that aerospace industry also we have a lot of opportunities. So aerospace industry, they are taking now chemistry people. And other than that, any field you can move forward. But these are the hot topics at this moment. Just doing the PhD in chemistry, you can very easily move to the aerospace industry, pharmaceutical industry, chemical industry, agrochemical industry.
All right. Thank you. Another question. Can I ask another question?
Sure.
Yeah.
That's why I kept my PPT short. I would love to give more answers because it's the practical aspect of life.
Yeah. What are the scope of PhD scholars in organic chemistry after completion in pharmaceutical industry?
Sorry, can you repeat one more?
What are the scope of PhD scholars in organic chemistry after completion in pharmaceutical industry? After completion in pharmaceutical industry.
So I hope the question is that after PhD or in organic chemistry, what is the scope in pharmaceutical industry?
Yeah.
Yeah. So you have all the scope. That's why I explained that you have a synthetic, which is actually pharmacy means what? Any medicines. Medicines may have two parts. We can call it like you can see very easily Dolo 650. Everybody is using Dolo 650. What is Dolo 650? It is simply paracetamol. So paracetamol is actually active ingredients. We call it API, active pharmaceutical ingredients. So doing your organic synthesis in pharmaceutical world, you can easily involve in the synthesis of active pharmaceutical ingredients. Then you can move to the other perspective. Organic synthesis, after sometimes you can move to the regulatory strategy, which I explained that ultimately the regulatory quality. We can move to the statistics, which is quality by designs. And ultimately, if you want to go to the innovations, obviously there is a lot of chance you can go to the structural biology, you can go to the screening aspect, you can go to the stimulus. So that's why I use the word. Whatever the passion you have a lot of opportunities in pharmaceutical industry, but it depends upon what is your passion. Which, if you don't like something, it is better not to pursue. But if you like something, just move forward.
Thank you so much. And I expect this question asked by Kaustav Banerjee, who is our ex-student from President's University. And thank you so much, Dr. Haldar. I now transfer the call to
Hello, am I audible?
Yes.
Yeah.
Okay. Thank you, Dr. Majhi. I would like to thank Dr. Pranab Haldar for your thought-provoking speech and such an excellent presentation. And I'm very sure that our students will be very much benefited by your speech. Thank you also, Dr. Pranab Haldar, for chairing the session. And now we are at the end of the webinar. And I would like to request Dr. D.P. Chatterjee, the joint convener of this webinar, to deliver the vote of thanks.
Thank you, Dr. Bose. So by this time, we have reached nearly to the end of today's program. Hope all of you have enjoyed all three illuminating lectures. On the behalf of the organizing committee, I would like to thank Professor Dipanjan Pan, Dr. Vishnu Joshi, and Dr. Pranab Haldar for delivering lectures to this platform, making time from their extremely busy schedule. We'd like to thank Professor Anuradha Lohia, Honorable Vice Chancellor, Presidency University, for her gracious presence and presidential address that has in every sense set the tune of today's program. We would also like to thank you, Madam, for your constant encouragement and support for hosting such programs. We'd like to thank all the participants around the globe who have joined through Google Meet or YouTube and made today's program a grand success. We would also like to thank Google and YouTube for providing us the necessary online platform. I'd like to thank respected head of the Department of Chemistry, Professor Gandhi Kumar Kaur, for his invaluable suggestions, advices, support, and active participation in today's program, including chairing a session. Thanks are also due to Professor Bijan Dash, Department of Chemistry, for his advices and suggestions during the preparatory stages for today's program, and his active participation by chairing a session. We'd also like to thank Dr. Arnav Haldar, Associate Professor, Department of Chemistry, for chairing a session. Thanks are also due to the members of the organizing committee and every faculty member of the Department of Chemistry, Presidential University, for their support. Finally, I would like to thank both of our coordinators, Dr. Aditi Bose and Dr. Sasikumar Palani, for their untiring effort since the day of inception by taking care of every necessary point, without which we couldn't have executed today's program. With these words, paying our deepest homage to Dr. Sarvapalli Radhakrishnan once again on this special day, I would like to announce the conclusion of today's program. Thank you very much for joining and staying with us.